Hello, and welcome to this podcast for genetics. In this podcast, we're going to talk about reproductive cloning and therapeutic cloning. Now, these two topics are quite controversial, and so what I want to do in this podcast is just to explain some of the facts about the two and how they're done. It'll be up to you to decide how you feel about each of these types of technologies. My personal feelings on them, I will not share because I think it's important for you to investigate and to learn more about them and to come up with your own opinion. But let's talk about them in the sense of what they are and what they could potentially be able to do. Reproductive cloning, the purpose is to create an exact copy of an organism. Scientists have been successful at reproductive cloning with many different plant species and with many different animals. Some of the potentials for reproductive cloning: breed specific animals or plants that have features that certain individuals are looking for. Maybe animals that produce more meat, or plants that are larger and more ripe and tastier. You could also potentially make unique organisms. I don't mean like science fiction unique. I mean maybe a cow or a sheep that produces a particular kind of medicine in their milk, or a plant that produces a particular kind of nutrient that is not normally found in that plant. One could also imagine using this technology to repopulate endangered species. I'm sure you can think of some pros and cons of each of these, and I'll let you come up with those on your own. So let's talk about therapeutic cloning. And as we'll see on the next whiteboard, these two types of cloning are very similar, at least in regards to how they are done. But here, let's just talk about the purpose of therapeutic cloning. The purpose of therapeutic cloning is to harvest embryonic stem cells and not to make a whole organism. These embryonic stem cells, once harvested, they could be used to make various tissues, even whole organs. And this purpose is of clinical significance. How can they be used for a medical purpose? Some of the advantages of potentially making tissues or organs this way is there is less potential for rejection. Anytime an organ or tissue is used during a transplant operation, there's always a fear of rejection, and the patients have to spend the rest of their lives typically taking anti-rejection medicine. In this case, they would not have to do that. We could spend a lot of time talking about the benefits and the dangers of both of these techniques. But again, I'm going to let you do that investigating, and we'll also take some time in class to talk about some of these benefits and dangers to both of these types of techniques. But I do want to explain to you how these kinds of clonings are done. First, let's talk about reproductive cloning. Though there have been advances over the years, I'm going to describe the basic technique of something called somatic cell nuclear transfer. And to talk about this, I'm going to talk about sheep, similar to the sheep that were done in the original studies when the clone Dolly was made. So this is my poor attempt at drawing a sheep, but you're used to that now. And so let's draw a blue one down here. You will miss these fabulous drawings. All right, so what they do is with this red sheeps, so to speak, the real studies weren't done with red and blue sheeps, but this is a good way to represent them here. What they did was they removed an oocyte from this red sheep. And initially, this oocyte contained the haploid genome of this sheep. What they did was remove that nucleus so that we had an oocyte without a nucleus. But it's important to note that even though it lacked a nucleus, it contained all of the RNA, all of the mitochondria, and other organelles inside of the oocyte. The only thing that was removed was the nucleus. They then took this other sheep and they took a cell from the udder. And since that's a somatic cell, it is a diploid nucleus. They removed this diploid nucleus, and the denucleated udder cell was discarded. They then placed this nucleus, in fact, let me outline this in blue so we can remember it came from our blue sheep. They took this nucleus and placed it inside of this oocyte. Then, using electric or a chemical shock, they stimulated this oocyte with the now diploid nucleus to divide. So this is really important to think about. What they really did was they tricked this oocyte into thinking it was a fertilized cell with 2 copies of each genome. And so now it started to divide, and it went through several divisions until ultimately it made an embryo that looked like this. You have this clump of cells here in the middle. These individual cells don't look different from each other at this point. They're all looking pretty much the same. We call this the inner cell mass. This is what will become, or what is now, the embryo. These cells on the outside, which we can't really see, but these cells on the outside here are still important. They will go on to make things like the amnion, the umbilical cord, the yolk sac, other very important things, but this will go on to become the embryo. They now take this embryo and place it back into our red sheep, back into the uterus. And now this embryo is allowed to divide and grow and develop until, at some point, this red sheep gives birth to this cloned sheep. And this cloned sheep is identical to this sheep here, because it is the diploid cells that came from here to make this one. Now, some things I want you to keep in mind. The reason this was so unusual and such a landmark study was that they used, or they could have used, any cell from this sheep. They didn't use reproductive cells, just any old cell from this sheep to carry out this process. So what is therapeutic cloning? Well, remember I said therapeutic cloning is not the purpose of making cloned organisms, but rather a clinical purpose. So let's consider therapeutic cloning here. Now what you'll see here is I'm using the exact same picture. Essentially the same thing happens. We take it all the way up to this point here, but instead of transferring this embryo back into the red sheep, so what they do is they take this inner cell mass containing these embryonic stem cells and remove them from this developing embryo, and they grow them in a dish. And so now what happens is you have this plate of embryonic stem cells. They can now use these stem cells because they have a larger understanding now of how to treat them to produce certain cells, certain tissues, certain organs. Organs are a little harder to construct, but certainly using these stem cells from this developing embryo can now be used therapeutically. Now I kept this drawing with the sheep because really this could be done with any organism. One could imagine that we could use humans in this situation, where you'll take a donor egg and remove the nucleus, just like we did before. And over here you have another human, where you get their diploid nucleus from. That then goes into this oocyte and goes into this stage here, and you harvest the embryonic stem cells. And now what happens is that these cells, these embryonic stem cells that are growing, are genetically a match to this individual here. So for instance, if this individual needed new skin cells because of a bad burn, or in the future if they needed a new kidney because of a damaged kidney, one could imagine growing skin tissues or kidneys using one's own DNA through this process. And by using that, there would be less of a chance of rejection because it is their own DNA. Now I should say that right now, somatic cell nuclear transfer can be simplified. In fact, what we've been able to do is this. We've been able to eliminate the donor. We've been able to take a cell. I guess humans don't have udder cells, so should we get rid of that? But we've been able to take a skin cell from this person here and stimulate it to divide and produce embryonic stem cells. And though you don't need to know the specific genes, but essentially there are 3 genes that need to be overexpressed to make any cell in our body think it is now an embryonic stem cell and begin to make embryonic stem cells. If you want to learn more about this technique, take an advanced genetics class, molecular genetics or epigenetics, and you'll learn a little bit more about this particular process. All right, that is all I really have to say about reproductive and therapeutic cloning. We'll talk more about this in class. If you have any questions at all, please make sure you come see me. If not, I will see you in class. Bye.